Marine enzymes. The global enzyme market size was over $7 billion in 2015 and is estimated to exceed $10 billion by 2024. Enzymes serve as useful biocatalysts for several industrial processes and chemical reactions. Application of enzymes minimizes the cost and reduces the time of manufacturing processes. They are used in production of food and beverages, detergents, biofuel, in pharmaceutical industry, and in diagnosis of diseases, but also in research and genetic engineering. A variety of enzymes has been isolated from marine bacteria, actinomycetes, fungi, fish, and many other marine organisms. The biotechnologically important marine enzymes can be classified into proteases, lipases, amylases, chitinases, agarases, carraginases, and dienases. Due to habitat-related characteristics such as salt tolerance, hyperthermostability, barophilicity, and cold adaptivity, marine enzymes are relatively more stable and more active than the corresponding enzymes derived from terrestrial plants and animals. They possess novel properties and have features desirable for biotechnological application. Proteases, the enzymes that degrade proteins, comprise the global sales of more than 65% of all enzymes. They show broad pH and temperature activity and stability range, resistance to surfactants, bleaching agents, and organic solvents. Among different types of marine proteases, the alkaline proteases are the most commonly used. These enzymes are active at alkaline pH and temperature from 30 to 80 degrees Celsius. Due to their unique features, marine proteases found application in different branches of industry, mainly in detergent industry, food industry, pharmaceutical industry, and textile and leather industry. The thermophilic protease called alkalase was isolated from Bacillus subtilis, but for commercial use it is produced by Bacillus ligeniformis. The enzyme is active at the temperature of 60 degrees Celsius and at pH of 8.5. Alkalase is commonly used in the food industry for the processing of soy meal and protein-fortified beverages. The application of the enzyme as anti-fouling agent to remove bacteria was tested. The activity of another marine protease was discovered by coincidence. It was observed that salmon hatchery workers who salted salmon eggs had smooth skin on their hands. Later, in salmon sala, scientists at Bergen University identified a zonase enzyme in fish eggs. The enzyme helps the larva getting out of its eggshell by digesting its protein structure. Besides the zonase enzyme, the hatching fluid also contains other proteins. The mixture of compounds from salmon hatching fluid is sold by Aqua Biotechnology as Aqua Beauty with various skincare properties. The anti-wrinkle activity is the most potent effect that can be observed. The extract contained unsaturated fatty acids, proteins, DNA, RNA, vitamins, and minerals. The microorganisms living near hydrothermal vents have unique enzyme systems which let them survive in these harsh conditions. Some of the enzymes are of significant application value in molecular techniques. For example, from a hydrothermophilic archaea, Pyrococcus furiosus, a thermostable DNA polymerase was isolated. The enzyme has polymerizing and proofreading activity even at 100 degrees Celsius. fDNA polymerase is applied in high-fidelity polymerase chain reaction experiments. This application include cloning, gene expression, and site-directed mutagenesis. Amylases is another group of important marine enzymes. Among others, they have wide application in sweetener and beverage alcohol production. Fuelzyme is a brand name of the extremely thermostable alpha amylase isolated from Thermococcus living near a deep sea hydrothermal vents. Verenium Corporation developed the production of liquefaction of starch-based mushes and slurries during ethanol production, releasing dextrins and oligosaccharides with better solubility and with low molecular weight. The enzyme operates in pH range of 4 to 6.5 and temperature above 110 degrees Celsius. The enzyme breaks the internal alpha-1,4 glucosidic bonds in starch and its degradation products. High production cost is a major factor that restricts the growth of marine enzymes market. The other problem is that microbial enzymes are immunogenic, so they can behave like allergens. Therefore, before adopting a new technology, every care and precaution should be taken to safeguard human health.